is your super report from Channel 11. We're here at the Center for Tissue and Cellular Preservation. We're going to be interviewing Professor Oliver Trejo. He is an expert on cellular and tissue preservation and mummies too. He's going to be giving a conference on the subject. The conference is about to start. Could you please tell us what your research is all about?
My research is cellular preservation. So first I had to learn types of cellular death. This and that top one there. Then I had to learn how we preserve it and how another way to preserve it. And then I learned really how to make a mummy. That I did this project is because I like Egypt and I like its mummies. And mummies are preserved. This one isn't really preserved. The first one is apoptosis. Apoptosis is programmed cell death, PCD. The nucleus breaks and the cell changes. This is apoptosis. So this is an embryo hand, pretty much a shapeless blob. Then these aren't growing. You know how it made the blob? Where the cells that were here, and here are dying. And then they're dying even more on this, right here. And here is my hand, right there. And this is an actual picture of apoptosis. And also, the nucleus breaks when these cells die. The cell die here is, so this, pretty much the life cycle of a cell, though not the best part. So this is the normal cell. This is when it's getting smaller and stuff. This is when it's breaking up. This is what's left after it breaks up. This is the last piece of the cell. This is a phagocyte cell. It eats the little cell. To continue about cellular death, there are three more types of cellular death. Necrosis, conification, and entosis. Necrosis is when the cell dies from injury. Like when you cut your hand, like a paper cut, say. The cells that you just cut open are dead. 3 is conification. Conification is a type of cellular death on the top layers of the skin where they die and fall off. 4 is entosis, also known as cannibalism cellular, seen in some types of cancer, a cellular disease.
Professor, could you please tell us about human decomposition?
Okay, so this is human decomposition. There are 5 steps. The first step is fresh. It starts after 6 hours after the heart stops beating. After 6 hours, muscles become rigid, rigor mortis. Cells lose their unity and break down. This is known as autolysis. Second is bloat. Microbes multiply and gases expand. The gases produced cause natural liquids and tissues to become soft and break. 3 is active decay. The greatest mass loss occurs in this stage because of feeding of maggots and fluid loss. The 4th is advanced decay. Decomposition ends. And then 5 is dry remains. All that is left is dry skin, cartilage, and bone. My main topic is cellular and tissue preservation. There are 2 ways. One is cryopreservation, by cooling to low sub-zero temperatures. Here cell death is stopped, but needs special solutions for the cells not to be damaged during freezing or warming up. Examples: eggs, sperm, blood, plants, and embryos.
So, Professor, what is cryopreservation useful for?
It's important that you can even preserve embryos for the future. The second one is natural preservation. Mummies that are naturally occurring because of environmental environments, such as extreme coldness, acids, salinity, or desiccating dryness, have been found all over the world. Many corpses, so-called bog bodies, have been found in bogs in northern Europe. Okay, a mummy is a preserved body of a person or animal. Facts: any dead body that still has skin on it after a long time is considered a mummy. Types of mummies. There are two types of mummies: natural mummies and artificial mummies. Natural mummies are accidents of nature, which means that when they were buried, they turned into mummies because of the hot climate. The natural mummies that I'm talking about, and this are in Egypt. They're also for the desiccating hotness in the natural preservation. And how to make a mummy. Preparing the body. The body was washed and an incision was made in the abdomen to remove organs such as the lungs. The brain was removed through the nostrils. The brain here is being taken out from the left nostril. So they're taking it out with a long iron hook and putting the mashed brain in a bowl. We'll throw it out later. Organs for organs. The intestines, lungs, liver, and stomach were washed and covered with salt, natron, to dry them out, preserve. The heart was not removed. 30. Alabaster chest. Alabaster is a type of stone. Organs were placed inside these jars for the gods' protection. 4. Preservation of the body. The body was covered in natron for 40 days to dry the moisture out, and then it was stuffed with herbs and sand and covered with oil and perfume. These are the canopic jars that I was talking about for the alabaster chest. There's the jackal, the falcon, the baboon, and the human. Wrapping the mummy. This can take up to a week, using linens by the head and neck, then by the hands and feet, then the rest of the body. Some amulets were placed between the wrappings. 6. Sealed for eternity. After the wrapping, the mummy was decorated with a golden mask and paintings. Later on, it was placed inside a coffin, then inside a second coffin, and finally inside a stone sarcophagus. These are kind of the steps of mummy making. So this is the wrapping right here. This is placing it inside the first coffin, then the second coffin, and finally in the sarcophagus. And this is a model of a mummy I made. So this is to show the wrapping, and this is its mask. And this is some liver I got and put it in this jar. It is now in the bloat part of the decomposition. We're trying to preserve it.
How are you doing that, Professor?
With the salt. We're covering it in salt and then letting it dry in the salt. If you want to take a close-up, you can. There. And then this is preserved muscle, also known as beef jerky. If any of you...
Thank you very much, Professor, for this very good explanation about cellular and tissue preservation.
It was my pleasure. Anytime.
That was Professor Oliver Treasure talking to us from the Cellular Tissue Preservation Institute. Thank you very much. That's all for tonight.